I suppose that most of us, at some time or another, have mowed a lawn and tipped the grass cuttings into the wheelbarrow and then decided that we've really had enough and we'll go into the house for half an hour to have a cup of tea and a bun and recover. The surprising thing is that when you come out of the house again, 2 hours later, you find to your astonishment that the grass in the middle of the pile of cuttings is really very hot. In fact, that's much hotter than it could possibly have got by just standing in the sun. Well, the reason for that is that in there with the grass cuttings, there are billions upon billions upon billions of tiny single-cell creatures, the bacteria, which are breaking down the cellulose of the plant stems for food and generating heat while they do it. And they don't have to be imported, they just come along with the grass cuttings, or they're in the air. In fact, the very same bacteria that are doing that job in the wheelbarrow are doing it inside a cow's stomach, helping it to digest the grass that it eats. And if it comes to that, if we kill the cow and eat it, then inside our own guts, there are more bacteria which help us to digest the food that we eat. Some bacteria have very bizarre appetites indeed. I mean, they live on the paraffin in aeroplane fuel tanks, and they'll eat sewage. And some of them will even eat rocks. Well, Professor David Hughes at the University of Cardiff has been studying bacteria now for most of his working life, and the very last thing he is bored.
I suppose you could describe me as being in love with them. I'm just curious about them in one way. I want to know how they grow and how they put themselves together and how they manage to eat all these strange foods. I mean, but at the same time, I'm interested in using them. We can use them and exploit them. We do that in the sewage works. We're very much involved with purifying sewage and so on, so that I'm both curious about them, and that's, you know, being a scientist really, I suppose, and I want to use them as well and to exploit them. And when they do nasty things, I want to stop them. We are absolutely dependent on them. If we wipe them out, if you use that stuff which kills all known germs, and bacteria are germs, we'd die in about 5 or 10 years. We couldn't keep alive. So important.
I mean, why? What would happen? You mean that?
Well, for instance, most plants couldn't grow without microbes. There are microbes taking nitrogen from the air and making it available to plants. There are microbes that take nutrients from the rocks and make them available for plants. Now, all flesh is grass, and we're dependent on plants, and the plants would die. And then, of course, microbes have a very important role to play in the natural cycles on Earth. They're the real decomposers. Anything that's formed on Earth and dies, a microbe can eat it, you see. So that nothing would decompose. And, you know, we're all dependent on death and decomposition and rebirth.
These are the signs of decomposition. Bubbles of methane, the natural gas that can be used for heating or for driving machinery. This is the University of Cardiff's experimental digester, and the researchers' aim is to improve its efficiency, mostly for the benefit of third world countries. But what they learn could help farmers everywhere. The research is supervised by Dr. David Stafford.
It's a plug-flow digester, and we've designed it not only for this country and Europe generally, but for the third world. And this material here is ensiled material. It can be grass or any kind of... Well, what we've done is we've taken a crop or waste green material, put it in large bags and kept it over the winter. And this can be fed into the digester as it's produced acids and sugars in the bags, and can be fed into the digester along with any kind of farm waste. In this particular case, we've got pig waste going in here in a mixture with the ensiled material, and it goes under a lip here and into the digester here, which is a long tube. And as the bacteria start to break down the cellulose here and the pig waste, then gas is released. And this gas is collected through this pipe, through a gas meter here, and the digested material goes through the digester, down a slope, and is collected in a pit at the far end.
Why does it go that way? You're not pumping it through?
Not at the moment, no. We're in fact, it's going through under gravity. And this solid material actually liquefies as it goes through the digester and so flows.
Right. So what have we got here?
we've got the treated material that's been through the digester, the cellulose has been broken down, the pig waste has been broken down, and we have basically a liquid containing the fertilizer that we had at the beginning.
Oh, so this has a value of its own. I mean, this system is not just to produce methane.
No, you have still the retained fertilizer value. You have no smell, so it can't harm the crops or can't harm people using it. It can go straight on the land through a pump. The fertilizer can be used by the plants, and the big thing, of course, is it has no smell.
So, in fact, you haven't, in a way, you haven't paid any price at all for the methane gas you've taken off.
Now, you've got the gas off, you've taken out the smell, and you've still got all the fertilizer value that you had at the beginning.
This liquid fertilizer will be used to grow more grass, which will feed more cows. They'll produce milk and more waste, which again the bacteria will convert to methane and fertilizer, and so on and so on. But how can something so small that it's invisible work such a transformation? Well, to start with, the bugs multiply so fast that there aren't just thousands of them, but thousands of billions. And what's more, think of almost any substance, and there'll be bacteria that can live on it. Bugs take in chemicals and convert them to what they need. Energy so they can move, or what they're made of so they can grow. And when they've taken what they want, they throw out what they don't. Those in the digester eat the straw, the pig waste, and any toxic chemicals that come in with them, throw out methane and produce a poison-free, fertilizer-rich fluid. Perhaps best of all, for a world that's running short of food, there are so many bacteria that the surplus can be dried off and turned into a high-protein food for pigs or hens, or even food for the cows themselves. And like the microbes that eat the grass cuttings, these bacteria are just there waiting to be invited.
Microbes live anywhere where there is a possibility of life. They'll live in hot springs. Those beautiful colours of hot springs are microbes. Well, they're bacteria. And they'll live in cold stores, live on glaciers, and so on. So they're very diverse. They have to be.
Hi-ho, it's home from work we go. Hi-ho, 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 hi-ho.
A disused copper mine in the heart of Snowdonia. Ian Kelso and Barry Johnson from the University College of North Wales have brought me here to see a unique example of that bacterial adaptability. It's one of the few places in the world where you can actually see bacteria with the naked eye.
Wonderful colours, aren't they, here? Ooh. Painful sharp.
Oh, yeah, that's it again.
There's precious little life down here, apart from us and the bacteria. They're not visible yet, but they're hard at work all round us, with sometimes alarming results. Ian Kelso showed me that any doubts I had about our safety were well founded.
Have a look at this crumbly rock here again, look. Solid rock. Oh, watch out, watch out.
Lovely. All right. Yeah.
Microbes again.
Microbes again.
The bugs involved are eating the rocks, or more precisely, converting iron in the rocks from one form to another. Their name is Thiobacillus ferrooxidans, but they're only a part of a whole bacterial society we're on our way to see.
This is the halfway stage in the oxidation process.
What's this stuff?
Oh,
Is that what it ends up
That's the end. That's the end stage. Ferric oxide, ferric hydroxide. The rust, the very end
So
stage.
it goes from the hard, glittery stuff we saw to that,
That's
and
right.
then it ends up as this.
Is that. And of course, the
yeah.
the process where the bugs are involved, the microbes, is from this stage to that one. Yeah. It's amazing, isn't it?
Isn't it just? How much further does this go on?
Oh, miles, yes, miles.
Further down the mine still. It's wetter, colder, harder work, and suddenly like the set for a horror movie. This glistening river of jelly is bacteria, living on the waste products from the iron-eating bugs. Each individually invisible, but in such numbers that they cover the rock face. Elsewhere, they hang down in rubbery stalactites. The jelly creeps down through the mine until it falls into and fills the underground stream at the bottom.
Why are the gloves a good idea, Barry?
Well, water is quite corrosive and it doesn't do your hands any good.
In fact, it's dilute sulfuric acid, which is the unlikely byproduct of the iron eaters.
Good grief, it goes down. It goes down forever.
A long way.
Good Lord. Oh, rotten eggs.
That was a mistake.
Right, well, thanks for that. I can't see it being one of the great tourist attractions for Wales, though.
Another part of Wales that doesn't attract the tourists. A quarry in Anglesey that was once another bigger copper mine until over-ambitious mining made the roof fall in. A hill became a hole, the bottom of it filled with more of that brown acid water. The pastel colours are traces of minerals, but the main ore, once rich in copper and iron, has been pretty well exhausted by the miners. By two kinds of miners, men and those same thiobacillus ferrooxidans that were working in the cave.
They were living on the pyrite, essentially on the iron within the pyrite. They were getting energy from the oxidation of it. They were eating the iron in the ore, essentially. And they were not using any energy from sunlight, as most living systems do. They were getting all their energy from this rock.
And this is how they left it, full of holes. They ate the iron and produced rust and sulfuric acid, which in turn dissolved the copper, so all that was left was a sponge-like stone. Meanwhile, the acid containing dissolved copper and rust found its way into the water that the miners were pumping out of the workings.
Bacteria have played a part in copper mining here, perhaps for 150 years, although no one realized it. The settlement ponds, which are constructed just around the edge of the mountain here, were used to collect drainage water brought up by pumps. The drainage water contained sulfuric acid, iron, and copper from the dissolution of the chalcopyrite, the main ore. The dissolution of the ore, of course, was brought about by bacteria, although people didn't realize it. They thought it was the action of air and water and so on. It was really the activity of bacteria that brought about this dissolution. And then they used the water from the pumps. They found they could collect copper from that.
Even today, throw in a lump of iron, the acid will dissolve it and copper appear in its place. It's a chemical conjuring trick that Barry Johnson, Ian Kelso's partner, demonstrated with a beaker full of fluid taken from the ponds. Before your very eyes, he'll produce 100% pure copper using only Ian Kelso's best pocket knife.
Yes, and you can see in that shorter time how copper has been deposited on this iron blade. This is a straight chemical effect. What has happened is some of the iron has gone into solution, and at the same time, copper has come out of solution and has been deposited on the blade. And this principle recovers the copper.
So this lake is acid enough to take your skin off because of the strange diet and even stranger byproduct of billions of bacteria. And even science found that hard to believe.
Well, the bacteria were discovered in 1947, and the bacteriological world didn't want to believe in them for many years. I suppose it was 1960 before they were really accepted.
Really, as recently as that?
That there were bacteria which could live in this way, generally accepted by bacteriologists.
Today, in places like Russia and New Mexico, 30% of the world's copper is produced by the action of bacteria. Bugs are at the centre of a very new technology. At Cardiff University, they're looking for bigger and better bacteria, finding ways to speed up their growth rate, to improve their appetites. It's a technology that's developing fast.
coming along now are the adaptations of our new technology of making bacteria produce other things, and this is what we call biotechnology. We can design the bacterium to make things which we are in great need of.
Most of our chemical needs today are satisfied by vast cathedrals of pipes and tubes that are expensive to run because they work at high temperatures and enormous pressures. A cheaper, safer, more adaptable answer could come from the versatile bacteria. But if you want them to produce a particular useful byproduct, you have to be sure that you've found the right one. A research team at Warwick University found their ideal bug in a very unlikely place. Heading the project, Dr. Howard Dalton.
This one originally came from Bath, in fact from the Roman springs there, where the hot water comes up out of the ground into the original Roman baths. And because the water comes up out of the ground at about 55 degrees centigrade, and because there's quite a lot of methane around there that comes up from underground, we started to look at a whole variety of sources of methane-utilizing bacteria. And we chose upon that particular one because of the high temperature. We wanted an organism that would grow at a fairly high temperature. And lo and behold, when we went down there, we got a sample of the water, we brought it back to the lab, and that produced the organism that we now call Methylococcus capsulatus Bath. So we started to look more at exactly how the organism could grow on methane itself. All the bug actually will grow on is methane. What it does as some sort of fortuitous activity is take on board all these other compounds and oxidize them, but not use the product. So it grows on methane, but it has the ability to take all these other compounds and introduce oxygen atoms into them and spit out the product, which is terrific.
Terrific, because their methane food is cheap and available. But give it to the bugs and they'll multiply like wildfire, producing a valuable chemical, propylene oxide, that's used to make a whole host of everyday products, paints, glues and plastics.
What we do is we grow the bug up on methane, we feed it propylene. The bug sort of can't really tell the difference between propylene and methane, so it just takes on board the propylene, makes propylene oxide. It can't recognize propylene oxide as something it can use, so it spits it out again. And so we have a system growing in which we feed propylene in at one end, and out the other end comes propylene oxide. The bugs are still growing all the time.
Yeah. So it's a chemical plant that actually grows. Every 20 seconds or so, each worker becomes two. And since that's true of all the thousands of species of bacteria, it's hardly surprising that they've become the basis of a new industrial revolution.
You've heard of the word biotechnology. It's very much in word at the moment. And then genetic engineering, which is designing microbes or bacterium for your own use. Actually, you combine the properties of several microbes. So you've made a new microbe or new bacterium.
The Centre for Applied Microbiological Research at Porton Down near Salisbury, which is interested in bugs, whether friend or foe. Its director, Dr. Peter Sutton.
That's right. I think biotechnology is here to stay, and I think there are two fallacies about biotechnology. People think either that it's extremely new, and in fact it isn't, because of course the use of microbiological systems for human good goes back a very long time. I mean, brewing is biotechnology, and the Babylonians were brewing 6,000 years ago, before Christ. The other thing is that it's extremely difficult to understand scientifically, and it's not. The science is really quite easy. Some of the terminology, some of the language is difficult, but the concepts aren't difficult.
There's an even bigger gap between these public health labs and the military side of Porton Down than the mile or so that separates them. The interest here is in overcoming any threat to human well-being. So the elaborate security measures aren't to protect secrets, but people. Shut away behind locked doors in specially ventilated rooms are the villains of the microbial world.
We've got some of the most unpleasant bacteria there are. Some of these are viruses, which are just small bacteria. So we have the small amount of smallpox work done in the United Kingdom is still carried on here. We have the diagnostic facilities for some of these new diseases which have come from Africa. Many people have heard of Lassa fever, green monkey disease, names that people have heard about. We're collaborating with the World Health Organization in evaluating the new vaccine against rabies, which of course is a terrible disease. We're also helping the Medical Research Council produce a vaccine against leprosy. So we've got some fairly unpleasant organisms here. We've got a fermentation pilot plant, which to all intents and purposes is a big brewery. But instead of brewing yeasts to make alcohol, we brew harmless bacteria, and from these we extract the substances which the bacteria can make. And of course, the substances that they can make are the enzymes. And the enzymes are the specific catalysts which the bacteria have, which enable the whole of what they can do. And indeed, the whole trick of living organisms is the possession of the enzymes that enable the living body to produce chemical reactions that we, up to now, have only been able to do with enormous pressures and enormous temperatures when we come to conventional chemical engineering. This really is at the heart of biotechnology, the exploitation of enzymes. Bacteria are looking all the time for things to break down to give them the energy they need. And if a bacterium has the ill luck to land on something that you and I would find very toxic, it sets about to find out how to use it. The other half of the equation, of course, is using the bacteria to make things that we want. That's building up rather than breaking down. And at both these things, the bacteria are very subtle and very clever. Because of the new advances in science, we can now change the bacteria to make things which hitherto they had never been able to make.
How much do you have to manipulate the bacteria to get them to produce what you want?
Can I give you an example of a product that is really priceless, which we're dealing with here? As you probably know, at the base of the brain is the human pituitary. It's about the size of a fingernail. Now, that little gland, it's a box of tricks, really. It makes all sorts of hormones that control our bodily processes. One of them is known as growth hormone. Now, some children are born lacking growth hormone. And unless they're given growth hormone, they'll finish up perfectly proportioned, perfectly intelligent, but dwarfs. Now, this can be cured by giving them human growth hormone. The catch is it has to be human growth hormone, because for reasons that are not properly understood, animal growth hormone doesn't work. How do we get hold of human growth hormone? There's only one answer. You've got to get it out of dead bodies. You've got to take autopsied pituitaries, post-mortem pituitaries, and extract the growth hormone from that. And without that, these children would remain dwarfs. Now, the science of genetic engineering has enabled us to get around that, because there isn't enough human growth hormone from autopsies. Obviously, it's a beastly business having to use autopsy material anyway. We now have the power to ferment an ordinary culture of these harmless bacteria, and as well as making bacterial products, silly old things if you like, make human growth hormone, because that's what their program tells them to do.
What the bioengineers do is to operate on the bacteria, not with microscopic scalpels, but with that group of natural chemicals, the enzymes. Bacteria have a sort of brain, the chromosome, and they also have sets of instructions called plasmids, which might, for instance, help the bug to resist antibiotics. The microbiologists can actually take the plasmid out of a bacterium and, using one group of enzymes, cut the circle of instructions open. Then they add the instruction to make growth hormone from a human cell and, using other enzymes, persuade it to join on to the open ends of the plasmid, so closing the circle again. Now the plasmid contains an instruction to make human growth hormone, and the scientists put it back into a living bacterium, which promptly sets about doing as it's told, making a desperately needed hormone.
This is the advance in the science of genetic engineering, genetic manipulation, call it what you will. Many people, it's an unfortunate name in some ways because it sounds as if you're doing things to babies, to humans. You're not. You're doing things to bacteria.
Bacteria can eat grass or rocks or poison. They'll produce gas and they'll make chemicals. They can become food for animals, they can rid us of pollution, or manufacture vital medicines. And yet, for all that, we're only just waking up to the value of these tiny life forms.
So this technology is just taking off. I have a dream that Sir Hans Krebs, my previous chief, who died last year, we felt that the next century is going to be the century of the biologists. The chemists and physicists have had their day, and now the biologists are coming in, you see.